Okay, Jojo.
It's good to go.
Yeah.
For those who doesn't know me, my name is Joshua Schengeler. You can call me Jojo. I'm from Cameron Lab here at Penn State in the BMB department. And today I'll be talking about fluorescence polarization technique. The outline, initially I'm just going to talk briefly, just a slide, about the history, then how the instrument itself works, what is the principle, what is the theory behind this assay, what are some of the advantages and disadvantages, what are the applications, what people have used it for. Then I'll give you some case studies, just 3 brief ones, but hopefully we'll be drawing some lessons from those case studies on what should we do and what shouldn't we do. Some special considerations before you start your experiment. Sorry, that went fast. Anyway, this fluorescence polarization is 89 years old. This is the first paper that was published on fluorescence polarization by Karen. And if you want to really learn everything about fluorescence polarization, this is the Bible. Craig has this book, and I borrowed it from him. So if you want to learn more, just let me know, and I can supply you this one. It is a really, really nice book. It just goes from scratch to, from the most simple scenario to really complicated stuff. So mainly, fluorescence polarization is used as a tool for studying molecular interactions, and I'll show you when I discuss what are the applications, protein-protein interaction, protein-small molecule interaction, and so on and so forth. Because it is taken in real time, many people think that all of the experiments that can be done with fluorescence polarization is equilibrium binding studies. However, by using more specialized equipment such as stop flow and others, you can actually do kinetic analysis to study the association and dissociation rates using fluorescence polarization. And again, we have those equipment if your research needed those stop flow and flow as well. It is a homogeneous technique, so it doesn't require separation of bound and free species, and this seems to be one of the major advantages of this technique, because some of the other studies, they require you to separate these 2 species, and then you analyze your data. And most of these techniques are actually by using radioactive chemicals, materials, so that's another advantage, as we'll be talking about those. And one thing is that fluorescence polarization is really being used as a workhorse for high throughput screening, especially in the industry, for finding small molecule inhibitors for whatever you're looking for. So this is as basic as it gets. So here you have your sample. You have a light source. Initially, your light will be highly depolarized, so you have in every direction light going. And in front of between the sample and your source, you have a polarizer. So you're basically choosing a certain wavelength in a certain direction. So you have vertical light going through your sample. Okay, once it hits the sample, depending on whether your probe is bound or not bound to a larger receptor, then this vertical light will be either highly depolarized or it will stay polarized. And again, once I'm explaining the theory, you will understand this vertical versus horizontal fluorescence intensities. And then this signal goes to the detector. And there's, again, a polarizer, because you just want to measure the vertical intensity and the horizontal intensity and nothing else. And this is the equation that is used to measure the polarization. And it is not actually measured in polarization, it's measured in milli polarization. And these values technically can go up to 1,000 millipolarization values, but experimentally people have seen that it doesn't really go more than 500. And if it is going more than 500, there might be some problem either with the equipment or your sample preparation or the way that you blank your machine. In addition to polarization, you will also hear the term anisotropy. Okay? If you had the polarization value, it is really easy to convert it to anisotropy value. Mathematically, they're related, so there is no difference pretty much. They share the same content information, so you're not getting more information out of one or the other. Now, how does it work? What is the theory behind this? So you have a small molecule that is fluorescently labeled. because of its small size and Brownian motion, it will be rotating, tumbling into solution. When this fluorophore is excited by plain polarized light, due to the rapid rotation, the emitted light will be highly depolarized. Now, if you think about it, if this molecule, now the small molecule is bound to a receptor, now the effective molecular weight goes up, the tumbling will slow down, and once you excite it with the plain polarized light, the emitted light will actually stay polarized. Well, of course, some of it will be depolarized, but you're basically looking at the ratio of the polarized versus depolarized, okay? And so this is the equation that describes the rotational relaxation time, because we are really looking at how fast this probe is rotating in solution, okay? And this rotation depends on multiple factors. It depends on the viscosity, it depends on the molecular volume, and on the temperature. And if you think about it, if you keep the viscosity and the temperature constant, so the only thing that you will be looking at is the molecular volume. And again, you have something very small, now it's binding to something larger. And that's why when we're doing these experiments, we try to keep our probe as small as possible and your receptor protein as large as possible, so that the change in the molecular volume is substantial, so that you can get a better separation of the bound versus non-bound forms. And technically, if it is non-bound, your vertical and horizontal intensities, they should cancel each other. So you have, as in previous equation, if these are equal, it's going to be 0, 0, or whatever is going to be 0. So when you measure the probe polarization value, it should be close to 0. But from experience, depending on the molecule, they all have their intrinsic properties. So some of them will be really close to 0. Depending if you're doing RNA work, you will see that these probe millipolarization values are slightly higher, towards 30 or 40. But again, it really depends on your molecule and the intrinsic properties of the fluorophore and whatever it's bound to. Okay. And again, I will go over it again. The last slide, I have all the considerations before you start an experiment, so this will come up again. Now, what are the advantages and disadvantages? The advantage is that it has a low limit of detection. All the work that I have done personally, I can go to really low nanomolar dissociation constant, but there is nothing that limits you to go into the picomolar range as well. The trouble really gets into if the interaction is very weak, then it becomes problematic, not just because you cannot do it with this technique, it's just that it requires so much protein that it's not feasible in, in most cases, just like yours, you cannot purify as much of your protein. So if the dissociation constant is really high, you're not going to be able to do it. Just it's not possible. As I mentioned again, you don't need to separate the bound versus the unbound form of the receptor. No radioactive waste is generated, so it's environmentally friendly. It's sensitive to variations in the probe concentration, and this is a very important point that I will come back to it again, is that it doesn't matter whether you use 1 nanomolar of probe or 0.1 nanomolar of probe. Your millipolarization value should be constant in each case. The only thing changing is the intensity value, but that shouldn't affect your results. But I'll come back to it again, because yes, you can go as low as you want, but you cannot go as high as you want. Okay? It's a rapid response. The equipment that I'm using, 30 seconds, that's it. For a single tube, of course. But I'll talk about the equipment that we have in our department as well. And some of the disadvantages is the combination of lifetime of the dye, size of the probe, and size of the receptor matters. So in some cases, you just cannot do it. If you're doing protein-protein interactions and they're similar size, it's probably going to be difficult to do. Yes, you can do it, but the resolution is not going to be high enough, or you won't be able to differentiate between the unbound versus the bound form, just because the molecular weight difference is not big enough. With small molecules, you can get a difference between your probe being 1 kilodalton, let's say, and your protein 30 kilodaltons. That's really good change. But if it is 1-to-1 change, from 20 to 30, you're not going to see a big difference. And that's why also it matters that you fluorescently label whatever is small and not the big compound. You can suffer from autofluorescence. That hasn't been an issue for me. so I cannot give you too much insight on that one. And the instrumentation, the older instruments, because they were single tube measurements, those equipment were relatively cheaper, but now it seems like they just included the fluorescence polarization capability into plate readers that can do a bajillion other things, so that's why the value is really high. It's really hard to get that kind of plate reader that would be dedicated for fluorescence polarization only. So the current equipment, they're relatively expensive. 50,000 and above, just give a ballpark. But again, it depends on the company, whatever else it can do. So these are some of the applications that people have used it for. You can look at protein small molecules, you can look at protein DNA, protein RNA, protein lipid, of course water-soluble lipids, protein peptide, protein-protein interactions. I'll give you some examples of kinase assay, phosphatase assay, and it was used for protease assays as well. Okay, so here's a case study number 1. Here's anti-fluorescein, so an antibody binding to a fluorescein itself. Okay, so this one is relatively simple experiment, right? You have your fluorescein, which is already fluorescent, and you have your antibody that is going to bind to that small molecule. So it's a simple binding. You titrate your antibody. Your probe concentration is constant, so you see this increase in fluorescent polarization. Initially, when you have no antibody, your millipolarization value is really, really low. But once you start titrating it, because the molecular volume is increasing and more of the probe is bound, now the signal goes up, and eventually it will reach the saturation level. Now, as I said, I'm not giving you just basic examples. I want us to draw some lessons from each of these case studies. As you can see here, there's observe and correct it. In some cases, right, when you have your protein and you have your fluorescently labeled molecule, what you will observe is that once you start adding your protein, the intensity value of the probe will go down significantly. Okay, and this is, in some cases, you're saying, it's fine, it's quenched just because it's bound to the receptor and it changes, emission characteristics, whatever. But if the change is really, really significant, then there might be something more going on. So there are equations that I'm going to supply you with this guide from my equipment that, it tells you if the changes are really, really significant, then you should use these equations to correct the values. So, so this is initially what this is what they initially observed, but once they correct it, this is what they get. So the lesson is that you need to pay attention to the intensity values going up or down.
Yeah, I was going to ask what if the fluorophore changes?
Yeah, exactly.
It's quenched or something.
Yeah. I mean, if the decrease is like from 10 to 5, that's not a significant difference. But if we were talking, 50 or 100-fold change, that is significant. Okay. Normally, when I'm doing these experiments, of course, if I have an idea of what the approximate KD should be, I try to go 5 times below the KD and 5 times above the KD, and this will really give you a reliable dissociation constant that you can extract from this data. Maximum value, you will not always get 400 something as a saturation point. This really depends on what your small molecule was and what was your protein as well, and what is the size of it, what is the size change. So, I have had some complexes where the saturation point was at 200, another complex at 300, so it really depends on what you're using in your experiment. So that's the maximum value. The minimum value, I mentioned earlier, it really depends on your probe. Some probes, such as the water-soluble lipids that I'm using, that one is really close to 0, and that's what it should be anyway, as close to 0 as possible. But we have some RNAs and DNAs that the initial millipolarization value goes up to 30 or 40, and that's normal. Yeah, exactly, because it's larger. In some cases, it has been that the millipolarization value goes to negative. That means you just didn't blank your machine well enough. Start over, don't overthink it, just start from scratch. Just throw away everything that you have. Okay? So the lesson is, watch the intensity value. So we talked about if the intensity value goes down, but what if it goes up? That I will mention in the end when we go over the whole list of what you should pay attention to. Okay, so this is case study number 2, estrogen competitor screening. So what they had here is they had a receptor. It was bound to estrogen, so this one is the labeled version. So you have a signal initially, but now you're competing it with the unlabeled form, so you would expect the signal will go down. So this is a competition assay, okay? You cannot always start from competition assay directly. You need to know the KD of it. So initially you will do the binding experiment, but then you will do the competition experiment, because sometimes what people worry about is, okay, I have my small molecule, it is fluorescently labeled, but what if the fluorescent tag is doing something to increase the affinity or decrease the affinity or whatever? So this is one way of, you know, competing with the natural form of this chemical that you're using, and just to see if the IC50, the inhibitory concentration that you're gonna get, is comparable to the KD that you were getting with the fluorescently labeled form of this. Okay, so again, there's a receptor, fluorescently labeled small molecule. Initially you have some signal, then you're competing with the unlabeled form of it, and as it replaces the fluorescently labeled one, the signal should go down. Okay. Normally, when I do these kind of experiments, I set my protein concentration at its KD when the protein is bound. 50% of the protein is when the protein is bound to 50% of the probe that is present in the solution. And why is this important? Let's say you have a curve, right? The binding curve, and the saturation, you reach saturation at 1 micromolar. If you do the competition experiment at 2 micromolar, you're already at saturation. You have more protein. You have most of the protein that is bound to all the receptors, but then you have some extra protein that is unbound to anything. So when you add the competitor, you don't know if it's really competing out or just binding to other unbound form of this protein as well. So when you're at the 50%, you're getting that really dynamic range where you can see the increase, decrease, whatever right away. Well, decrease right away. So that's why it's important to set the protein concentration at the KD. Was that clear? Now this is more honest until I looked at this guide in more detail. You know, these are the experiments that I've never thought about, but I thought that, you know, they're more complex, and, you know, it's just all these different experiments that you can do with it. It's not simply just binding or you know, competition with other chemicals to find small molecule inhibitors and stuff, but people have used it for, you know, to detect if there is kinase activity, if there is phosphatase activity. So how does this assay work? Let's say you have a substrate. Let's say it's a, you know, it's a peptide. So you have serines and threonines and tyrosines that can be phosphorylated. So you can have your kinase that is going to phosphorylate this one, okay? But then you can have an antibody that can detect only phosphorylated form of this peptide, but not the unphosphorylated form. So if you have a fluorescently labeled peptide, when it gets phosphorylated, the antibody will bind to this, and now your signal will go up, okay? Because this is the complex that you're forming. And again, this is just a different scenario as well. You can have both labeled and unlabeled, but, you know, the simple idea is that you can have a labeled peptide, you can phosphorylate it, and add an antibody that can bind to this phosphorylated form only. And phosphatase assay, of course, would work just the other way around, just by using an antibody that can detect the unphosphorylated form and not the phosphorylated form.
Jojo, can we get back for a second? So have you ever heard about an experiment like this? It's kind of a corner case, but so say you had a protein that's not necessarily that large, that potentially exists relatively in random coil when unphosphorylated.
Disordered,
yeah.
disordered, yeah.
So it's probably going to have like a high radius of gyration. That's
Sure.
the word, right?
Yeah.
But then upon phosphorylation, if it then switches to a more tight conformation, the radius of gyration will reduce unless you see a change in polarization. Have you ever seen any kind of study like that? It would have to be very specific to, you know, a protein that does something exactly like that.
Yeah. I mean, we do have.
You see what I was getting at?
Yeah, no, I know, yeah. So initially you have something that is.
Yeah, you have to change the shape.
It's kind of large, but then, you know, once it's phosphorylated, it gets maybe like more compact or whatever. I personally don't know anyone that have tried that, because, you know, that's simple enough that you can do with DLS as well.
Ah, that's a good point.
So, I mean, I know, you know, Taylor in our lab, she studies intrinsically disordered domains, and yeah, that's what she does. She just does DLS in order to look at the structure, and it really depends on the fluorophore. It's just too many variables that you need to consider before doing something like that, and the change should be significant.
Yeah, it would only work, in a very ideal scenario.
Yeah, and it might, and whatever works for one protein might not work for the other protein as well. Yeah, so I don't know any example, but if I was going to do that, I would probably use DLS. That would be simple enough, because then you will get a distribution that's something wider to something like really compact distribution. All right, I think this is like the hallmark that should be out of this presentation, because let's say you want to start an experiment. What are all the variables that you need to think about? But let's go step by step. First of concentration is the equilibration time, and actually there is no reason that you should go one by one, step by step. I really put them as they came into my mind, so there is no order to this one. But one of the important ones is the equilibration time. Okay, how long does it take for this molecule to bind to this molecule? And it can vary from case to case. Normally, if you're doing DNA, RNA binding studies, these are instantaneous. Okay, you just need to mix them and put it inside the equipment to do the measurement. But this equipment that I use, there are two modes that you can use. You can do static measurement. That means you put one tube, it measures it once, and it's done. Or you can do a kinetic measurement, where you can put this tube and let it do the measurement as a function of time. So you will say measure every 5 minutes or every 1 minute, and then you will get these millipolarization values as a function of time. And if the binding is dependent on time, I mean in a longer period of time, then you will see as a function of time that this will reach a certain saturation level, and you will then choose whatever the minimum time is required to do all your subsequent measurements. And normally, when I do this kind of thing, I would try to pick at least 3 concentrations, one at the low end, one around the KD, and one higher concentration. But of course, it's difficult to say that because you don't know what the KD is. But if you have some kind of idea, if you're doing DNA, RNA work, you know that it's going to be at the nanomolar range. If you're doing some lipid work or protein is going to be micromolar towards millimolar. So you have some kind of idea which protein concentration to start with. Okay. Another one is temperature. Normally I do it at 25 degrees. I mean, in my case, I can do it at 37 as well, but I have done both and I don't see a change. So I keep it at the lower temperature just so that I keep everything stable, but there's no reason to it. If your experiment requires you to do it at 37, do it at 37. We can go, with this equipment, I think we can go up to 60 degrees Celsius. So you can choose whatever temperature is necessary. As I said, the size is important, so your protein, the receptor should be large, and your fluorescently labeled ligand should be small. Normally, if you're going to have a ligand, I would go as high as 10 kilodaltons, okay? If you're using a fluorescein tech. If you're using fluorescein tech, the time from excitation to emission is 4 nanoseconds, okay? And for a 10 kilodalton protein to change its rotation from 0 to, I don't know why they say 68.5. So the way that the detector is oriented is at the 68.5 degrees. It's not 90 degrees. Say it again?
Is it like 1 radian, 2 radians, something like that?
I cannot disagree.
It sounds similar to a radian measurement, so it's probably consistent.
Yeah, it might be the case. So basically, the time that it takes for the protein, 10 kilodalton protein, to go from 0 degrees to 68.5 would be 4 nanoseconds as well. So if you're outside of this range, your light is probably going to stay polarized because you didn't have enough time to rotate to begin with because of your size. So that's why you need to be at least 10 kilodalton or below for your probe. Protein concentration range, when you're doing simple binding experiments, as I suggested, it should be 5 times below the KD and 5 times above the KD to get a reliable dissociation constant, at least from my experience. That's simple enough. Purity and contaminants. This is really important. There was this one study they had boatload of their protein. They showed an STS page, gel. Right? Their protein was boatload in there. There was a slight, this additional band that was, if you didn't print it in color, like you wouldn't even see it there. But then they realized it was such a high affinity to that low protein, that additional band, that everything that they got was just basically due to that contaminant.
That happens more than you think.
Yeah, exactly. So it's really important that you get something pure. And if you're having a reproducibility issue, it's probably that every time you're purifying, the amount of contaminants that you're bringing is just varying, and so you just need to figure out how to purify your protein better.
You ever run into problems with ligand impurity?
No, because these ligands are they're synthetic, so honestly, because the concentrations that they supply is so low, it's even hard to run a gel or anything in order to look at the purity of these. So if you have RNA, I'm doing my experiments like 0.4 nanomolar, and the stock that I have is, I don't know, 50 nanomolar, 100 nanomolar. It's really hard to run a gel to see that. And if I can do that 50 nanomolar maybe, but I'm going to waste all my probe.
Well, I was thinking, I have some RNA that was fluorescein labeled, and there's a whole lot of free fluorescein. So fluorescein sticking to the protein, that's going to dramatically.
Sure.
Well,
So
you have free fluorescein too. That would, if there's a bunch of it around, that would depolarize.
sure,
It's
absolutely, yeah. So
yeah.
you need to better separate this. So you can do you can run a gel and just separate it. Basically, you run one of the long gels, and of course, if it is labeled, it's going to run higher. And you just look under the UV light and just cut out that portion and just purify your DNA or RNA. And that has to be done. Otherwise, that is a problem, especially if it binds to. If the fluorescein binds to protein and you have potentially unlabeled form of DNA RNA there as well that can bind to protein, now you're not really, you get it, yeah.
Well,
you need to better purify your
Yeah,
protein.
have you ever run into that problem, or do you always take precautions to clean your probe off?
I mean, we always get it from Dharmacon.
Yeah.
We do most of the RNA work, and we say, give me a 5' or 3' labeled RNA with this sequence, and that's what they supply. And there's a purification method that you want them to do it as well. So, but normally, so every time you prepare one batch, I'm not talking about after a year your DNA or RNA might have degraded. You need to check the purity. You get the probe. You can run one of the sequencing gels or whatever and just look at it. And you can do it. Let's say if your RNA, for example, is 3' labeled, right? You can do 5' alpha UTP or whatever. Labeled phosphatase assay. So you can basically label the 5' end, and then you can see where your RNA is on the gel. But the higher one that shows a signal will also have a fluorescent signal as well, and then you can cut out that portion or whatever. Or at least just to see what is the ratio of the unlabeled or degraded form versus the fluorescent labeled. So there are ways that you can look at it, but it really depends on the cases. But that has not been an issue for me. Yeah. I mean, we have so many probes in the lab, and in cases we have reordered some of the things and we get the same data. So that has been the case. All right. Another thing that you should think about is the dispersity of your protein. Let's say you have a simple experiment. You have your protein and you have your RNA or DNA that you're gonna test the binding affinity between each other. You want your protein to be monodispersed. Of course, depending on the case, there are some proteins that are, binds to each other, and there's nothing you can do about it. But you don't want it to fully disperse, meaning they start aggregating. Okay, and the best way to look at it is just using DLS. You can look at it if your protein appears as a monomer, maybe dimer, and you can get the approximate weight of your protein just by looking at the DLS. But if you see multiple distributions, that means you just haven't purified your protein well enough, or the conditions are not appropriate, or you should just filter your protein. And that is what I have done as using these really small filters, just to make sure that everything that I had is in a bondable form, so that the results that you're getting is because the protein was bound to the RNA, and not just because protein randomly aggregated on top of the fluorescently tagged DNA RNA, and you're getting this random signal.
Usually use cellulose acetate filters.
Yeah. And these are like, how big? I think they were like 0.05 or 0.02 microns. Yeah, like really, really tiny, but again, really depends on the aggregation too. If you have too much aggregation, your solution just wouldn't even pass through. It's really, yeah. So you might need to do multiple filtering depending on the case. Buffer composition is important. So if you came to me and you said, Jojo, I want to test, binding of my protein to this DNA. Buffer composition that I will choose for the experiment would be, I would ask, what is your buffer composition of your protein? Is it in tris buffer, in phosphate buffer, in Heaps buffer, whatever? That is the buffer that we're going to use. So, don't try to change around with buffers when you're doing these kind of experiments, because you don't know how would probe react to.
And basically, what you're getting in there is, the technique is relatively insensitive to the buffer.
It is very sensitive.
Oh, it is sensitive.
It is sensitive.
Especially to salt.
Yeah. It is very sensitive. Some probes will be very sensitive to cations, divalent cations. Some will be sensitive to salt. Some will be sensitive to light. Some will be sensitive you looking at it. You never know. It is pretty sensitive. Again, call factors. Are there any divalent cations that is required for function? Are there any other proteins that requires binding to have that function? So all these things that you should have some kind of idea before you start the experiment. Probe concentration. Normally people choose 1 nanomolar, okay, and that is fine. But if you're thinking about doing a protein and RNA binding where the dissociation constant is going to be in the nanomolar range, now you need to be careful. Now why? The equation that you're going to use to fit a model that you're going to use to fit your data in, it doesn't have your complex in the equation. So you're basically assuming that the species, the complex that is being formed, is so small that you can exclude it from your equation, from your model. So that's why it needs to be as small as possible, so that you can reliably say, yes, I assume that the complex being formed is so minimal that I can exclude it from my calculations. Okay? And that's why if the binding is at the dissociation constant is in the micromolar range, then it is fine to use 1 nanomolar. Okay? Because now you've gone from micromolar to nanomolar, and that's, there's a lot of change in there. But if your binding constant is at 10 nanomolar and you're using 1 nanomolar of probe, you just need to be, careful when you're doing your calculations.
You're talking about a receptor equation, right?
Yeah. Yes, exactly. Yep. And as I said, because the millipolarization, the output that you're getting is not dependent on the concentration of the probe, you can just go as low as possible, depending on the case, as long as the results that you're getting are consistent. So basically what you can do is, okay, let's say I use 0.01 nanomolar. Take that tube, put it in, do the measurement. Take that tube out, put it back again. Are you going to get the same number? Or plus or minus whatever. Plus or minus 5 or plus or minus 10 millipolarization is fine. So yeah, you need to think about these things before you do it. I talked to you about lowering the intensity. So if there is binding, because some of the fluorophore will be quenched due to the changes in the excitation emission characteristic of the probe, yes, there is binding and that's fine. But if there is increased intensity, what does that mean? According to my experience, that has shown to be signal for precipitation. And in some cases you cannot tell when the intensity from blank to addition of the protein has changed by intensity value of 1 or 2. But once it's going like double the value, that is you are getting serious precipitation. And in some cases, I've taken the tube out and it just turned white. And that's when I knew that it is indicator of precipitation. And that's why I always be careful when my intensity value goes up. When it goes down, I'm happy. But when it goes up, I'm just, oh crap. Okay. When you're running your experiments, there always will be blank. In our instruments, you will have 2 blanks. One of them, all the buffer components, excluding the protein, excluding the probe. That's your initial, so it's only buffers. Your second blank is your buffer plus your probe. Okay, so now you have excluded all the diffraction that you're getting from the buffer components, and now you know what your probe millipolarization value is in the absence of any protein, and now you start adding your protein, and the starting value is whatever your probe in the absence of protein gave you, and then you start refrigerating your protein. So yeah, and that's why I said sometimes you get negative millipolarization value. That's probably because you messed up something with the initial measurement where you didn't have probe, you didn't have protein. You messed up, either you mispipetted or forgot to pipette or put extra thing. So that's where the error might have happened if you're getting negative polarization. One control to do it, to see if your protein is binding to the tag, is to do competition experiments and compare your IC50 value to the KD that you're getting and make sure that everything is making sense. Another control that you can run is, if you're if it is fluorescein tag, if you have fluorescein tag itself, you can test the binding to it and just see if you're going to see any binding. And again, where's the intensity change? All right, where's the equipment? So there are 2 fluorescent polarization capable instruments in 206 that I know. I don't know if anybody else has it, but if you have one of the near plate readers, you probably have fluorescent polarization as well. But we have 2 in 206 outhouse. One of them is dedicated single tube instrument, and that is the one that I'm using. But also we have another plate reader where you can do more high throughput screening with, I think 96 well plate is the highest that you can go. I don't know if you can do more. But not only fluorescent polarization, you can do a bunch of other.
You can just use standard plates, right? You use like a quartz.
That I'm not sure. That you need to check the manual. No, mine is these.
So you only do single?
Yeah, exactly.
Okay.
So these are just regular tubes. What kind of tubes? Borosilicate tubes.
Okay, so it's not.
Yeah, just simple glass. But yeah, this one, the plates. I'm pretty sure you need. I don't think that is just going to be normal plate, but I'm not. Don't take me on it. Just check the manual. We have the manual upstairs.
If you guys know, we have a quartz plate.
Yeah. Yeah, I'm just done with fluorescent polarization, so.
I'd imagine you only need the quartz for UV.
Yeah, who knows?
Visible usually isn't a problem. I mean, just have a glass cube and then it's usually the UV range. You have impurities of fluoresce, impurities in the glass.
Sure.
It's used quartz.
Yeah, I've never really tried that instrument for this purpose. So I cannot really say anything. I just know people have a problem with, so let's say if you have a 96 well plate, there might be some errors associated with the outermost wells. Yeah, I mean, so the advantage of doing the single tube experiment is when you're doing global analysis, right? If you're doing measurement all at once, if there is an error associated with the equipment, everything is going to have an error. Versus, if you're doing single measurement, every tube is a separate experiment, and combined data, then it will tell you if there was something wrong. But I think single tube is more reliable. But again, I cannot say that for sure. I just haven't had direct experience with it. It's just what I've heard about these plate readers. Anyway, again, the single tube instrument is easy to use, and it's really easy to train someone. You just need to tell them which protocol to use and how to handle stuff. So it's relatively easy, and there are not too many settings that you need to worry about. You can create your protocol. What temperature you want to use, how many measurements you want to use. Do you want to do static versus kinetic measurement for equilibration time? And that's pretty much it, and then you're set to go. So of course we need permission from Craig. So I mean there is a reason to it, not just because he's Craig, because the single tube instrument, and yes, everybody's going to watch this now, but because single tube instrument, this one, they're not made anymore. Okay, if you wanted to fix anything, there's no hope. Once the instrument goes down, it will go down forever. With the other plate reader, it's an expensive equipment, so we want to make sure that you're appropriately trained before you use it. But now the problem is that we don't have much experience with it neither. So Sun10's lab mainly uses that instrument. So normally when somebody needs training, we actually contact them and ask them if they can help us with the training of a person. So if you have more questions about that instrument specifically, you can actually directly contact them. I know Matt has used it a lot. So you need a permission from Craig. I have manuals for both, and one of them is actually, so all these case studies that I provided, they actually come from this guide that was designed for this single tube instrument and all the capabilities basically. So I can provide you that one. It's been very valuable. That one is a really, really good guide for everything. All these equations that you can use if you need to make any corrections to calculate certain parameters. It is very good. And if you have any questions, you can contact me. And as I said, I also have the Fluorescence Polarization Bible if you need it, so let me know.